Assalamu alaikum, welcome to VetLec. Today the topic of discussion is nervous control of gastrointestinal system. The gastrointestinal system is regulated by two control systems. One of these control systems is intrinsic nervous control and endocrine components which are located within gastrointestinal organs. The intrinsic nervous system which controls gastrointestinal systems is actually enteric nervous system of gut. So one control system is the enteric nervous system and endocrine components which are located in the gastrointestinal organs, and the second level of control is exerted by the central nervous system. So the control of gastrointestinal system through central nervous system is actually extrinsic control system, while the control of gastrointestinal system through enteric nervous system and endocrine components involves intrinsic control. In this way, coordination of gastrointestinal functions with the rest of the body is achieved by the integration of these intrinsic and extrinsic influences. The enteric nervous system is extensive and well defined and contains as many neurons as the spinal cord. Like nervous system, enteric nervous system also consists of cell bodies and their associated neurons, and all of these lie within the gut wall. Within the gut wall, cell bodies of enteric nervous system are arranged in two systems of ganglia. One system is known as myenteric or orbex plexus, and the other system is submucosal or Meissner's plexus. So, enteric nervous system is composed of submucosal neuronal plexus or Meissner's plexus in submucosa of smooth muscle cell, and second type of plexus is present in the muscularis externa between its longitudinal and circular layer, and this plexus is known as myenteric plexus or orbex plexus. So in short, submucosal plexus is present in submucosa, while orbex plexus or myenteric plexus is present in the muscularis externa between its longitudinal and circular layers. The function of submucosal plexus is to control the secretion of mucosal and submucosal glands. It also controls blood flow through the submucosa and lamina propria, and the submucosal plexus also determines the absorption. So in this way it regulates secretion, absorption and blood flow in this area, and it also controls, or it has influence on the neuroendocrine cells which are present in this area, means which are neuroendocrine cells which are present in submucosa. On the other hand, myenteric plexus is concerned with the regulation of motility of gastrointestinal system. Let me repeat it. Submucosal plexus controls the secretions of mucosal and submucosal glands, controls the blood flow through mucosa and lamina propria, and submucosal plexus also determines the absorption. In this way, submucosal plexus regulates secretion, absorption, blood flow in the submucosa, and also controls the cells which are present in submucosa. It controls the neuroendocrine cells which are present in submucosa. And on the other hand, the myenteric plexus is concerned with the regulation of motility of gastrointestinal system. The neuronal network of myenteric plexus and submucosal plexus is also intercommunicated by the individual neurons that leave the myenteric plexus and submucosal plexus. So these both are interconnected. These interneuronal connections within the myenteric plexus are extensive. On the other hand, these interneuronal connections within the submucosal plexus are limited. Due to this intricacy of the enteric nervous system within the gut, this enteric nerve it is also known as little brain. Different kind of neurons are present in the plexus of enteric nervous system, and these neurons are sensory neurons, interneurons, and motor neurons. The sensory sensory input which is passed to the sensory neurons comes from the mechanoreceptors which are present in the muscular layers, and chemoreceptors within the mucosa of smooth muscle cell. In short, chemoreceptors and mechanoreceptors stimulate the sensory neurons. The mechanoreceptors are present within the gut wall, and their function is to monitor the distension of the gut wall due to the presence of food material within the gut. So these mechanoreceptors monitor the distension of the gut wall. On the other hand, chemoreceptors are present within the mucosa of smooth muscle wall, and they monitor the chemical conditions in the gut lumen. These chemicals are actually different hormones that are secreted into the lumen of the gut, and then chemoreceptors detect these chemicals and then stimulate the sensory neurons. So both mechanoreceptors and chemoreceptors stimulate the sensory neurons. The other type of neurons that are present in the plexus of enteric nervous system are motor neurons. These enteric motor nerves supply the blood vessels in the smooth muscle, gut muscle, and glands within the gut wall. So, enteric motor nerves actually supply the glands which are present within the gut wall, the gut muscles, and the vessels which pass through the gut muscles. Due to the stimulation of these three structures by enteric motor nerves, enteric system actually causes contraction of gut muscles. The innervation of gut structures through enteric motor neurons doesn't occur through direct synaptic type of junction. Instead, the axons of the enteric motor neurons end in a tree-like structure of branches or end in arborizations, and these arborizations or branches contain many vesicular structures called varicocytes. So, in short, the gut structures are innervated by enteric motor neurons in such a way that the ends of axons of the motor neurons end in arborizations or branches or tree-like structure of branches, and at the end of each branch there is a vesicular structure called varicocyte. As I have said that coordination of gastrointestinal functions with the rest of the body is achieved by the combination of intrinsic and extrinsic influences. So the sympathetic and parasympathetic nervous system forms the link between central nervous system and enteric nervous system. Uh, in other words, in other way, we can say that sympathetic and parasympathetic nervous systems form a link between extrinsic nervous system and intrinsic factors and intrinsic nervous system. Most of the gastrointestinal tract receives parasympathetic innervation by the way of vagus nerve, but there is an exception in the terminal portion of colon, and it receives parasympathetic innervation from the sacral cord through the pelvic nerve. Neurons of sympathetic and parasympathetic nervous system influence the gastrointestinal functions through the release of neurocrines. In general, parasympathetic neurocrines are stimulatory. It means that they increase gut blood flow, motility, and glandular secretions, and in this way, they increase the gastrointestinal motility. On the other hand, the neurocrines of sympathetic system are inhibitory, and they try to inhibit the gastrointestinal motility. Now let's talk about the functions of myenteric plexus in peristalsis. When we eat food, then the food stretches the gastrointestinal walls. This stretching is sensed by the mechanoreceptors which are present in the gut wall, and these mechanoreceptors then stimulate the sensory neurons which are present in the muscle layer. These sensory neurons then carry the stimulus from mechanoreceptor to the outer plexus, which is myenteric plexus, and in this way sensory neurons stimulate the myenteric plexus. This stimulation of myenteric plexus can also be done by giving some signals to the mucosa. Let's suppose that there is a chemical signal to the mucosa which will be detected by the chemoreceptors that are present in the mucosa, and they will carry the information from mucosa from the chemoreceptors via sensory neurons to the outer plexus, and there can be some irritation to the mucosa, and this will also act as a stimulus for the stimulation of outer plexus. So myenteric plexus can be stimulated by mechanoreceptors, chemoreceptors, and to some irritation to the mucosa. And in this way, sensory neurons will take this information and they will stimulate the myenteric plexus. So these stimuli produce peristalsis, and these Stimuli cause the muscles of gastrointestinal tract which are present on the oral side to contract, while while the muscles that are present on the aboral side, these stimuli cause these muscles, aboral muscles, to relax. In this way, wave of peristalsis flows from oral side to aboral side, and contraction of gastrointestinal tract occurs from the oral side to aboral side. In the myenteric plexus, there are two interconnected layers of neurons, and these are which are present in the myenteric plexus. And one layer of the interconnected neurons is ascending network of neurons, and the other is descending network of neurons. The ascending network of neurons moves towards the oral side, while the descending network of neurons is a layer of neurons in the myenteric plexus which is moving away from oral side, or which is moving aborally. So, as on the ascending side of gastrointestinal tract, we want contraction, and on the descending side, we want relaxation, so that the wave of peristalsis will flow from the oral side to aboral side, or contraction will result from oral to aboral side. And in this way, we want stimulation of myenteric plexus on ascending side of the ascending network of neurons, or ascending side of the gastrointestinal tract, and similarly on the descending side of gastrointestinal tract, or on descending network of neurons, we want inhibition of the stimulation of myenteric plexus. So the sensory neurons stimulate both ascending neuronal network and descending neuronal network. So when the ascending neurons will be stimulated, they will produce stimulatory neurotransmitters, and these stimulatory neurotransmitters will stimulate the myenteric plexus for contraction. These stimulatory neurotransmitters are substance P and acetylcholine. So these stimulatory neurotransmitters will stimulate the neurons of the gastrointestinal tract that are present in the oral region. So these muscles will contract, and the contraction will occur in the oral region. Similarly, sensory neurons also stimulate the descending network of neurons. And when this descending network of neurons will be stimulated by the sensory neurons, it will start to release neurotransmitters which are inhibitory and inhibit the contraction of myenteric plexus. Sorry, it inhibits the contraction of smooth muscles. These inhibitory neurotransmitters are nitric oxide and vasoactive intestinal peptide. So these inhibitory neurotransmitters will be produced on longitudinal layer and some on the circular layer of smooth muscles. So circular smooth muscles will start getting relaxed, and this relaxation is receptive relaxation. In this way, the smooth muscles on the aboral region will start to relax. As on the oral side, circular smooth muscles are contracting, so bolus, and on the aboral side, the smooth muscles are relaxing, so the bolus will be pushed forward from oral side to aboral side. In this way, when the bolus will be pushed forward, it will again stimulate the sensory neurons in ascending and descending network. So ascending network of neurons will produce stimulatory effect and at new site, and descending neuronal network will produce inhibitory effect at that new site, and this process will continue the regulation of peristalsis within the gastrointestinal tract. So due to these contractions and relaxations of the smooth muscles of gastrointestinal tract, the bolus is moved from oral side to aboral side, and peristalsis occurs in this way. In the same way, parasympathetic nervous system releases neurocrines that are stimulatory, and sympathetic system releases neurocrines that are inhibitory. And in this way, both sympathetic and parasympathetic nervous system influence the gastrointestinal function or gastrointestinal motility through the release of these neurocrines. The gastrointestinal system also has an extensive number and variety of endocrine and paracrine cells, which are distributed diffusely throughout the gut epithelium. Endocrine cells are those cells that produce hormones that travel through the blood from their site of synthesis to their site of action. And on the other hand, paracrine cells are those cells that secrete the secretions, and these secretions travel by the diffusion to the nearby target cells, and in this way they exert their effect locally. Typically, both the gastrointestinal endocrine cells and gastrointestinal paracrine cells are columnar with broad base and narrow apex, and they both of these are present in the secretory and absorptive areas of the mucosa along with other mucosal cells. Their narrow apex of these cells is exposed to the lumen of the gut, and this allows them to sample or taste the luminal contents. This function is very important as this arrangement provides a chance for the cells to sense the changes in the gut luminal contents and then to respond by releasing their hormones and other regulatory substances. So the hormones and paracrine substances which are released by these cells, by the endocrine cells and paracrine cells, are actually stored in the form of secretory granules, and these secretory granules are present at the base of these cells. So endocrine cells secrete near the blood vessels, whereas paracrine cells secrete into the interstitial fluid. And some of the paracrine cells have long basal appendages, and these basal appendages direct the secretions of the paracrines to the vicinity of the target cell. The secretions of endocrine and paracrine cells have regulatory functions, and they don't undergo the process of digestion. There are many types of endocrine and paracrine cells which are present in the gut epithelium, and they all are morphologically similar. However, despite this similar appearance, many cells produce a wide variety of hormones and other regulatory substances. Most of these substances are peptides in structures and can be referred to as collectively called as regulatory peptides. And there are at least 28 peptides that have or that are suspected to have gut regulatory functions. For convenience and clarity, all these substances are frequently referred to collectively as neurohumoral regulatory molecules. Let me repeat it. Many different types of endocrine and paracrine cells which are present in the gut epithelium are, although they are morphologically similar, but they produce a wide variety of hormones and other regulatory substances, and the secretions of these endocrine and paracrine cells are regulatory and not digestive. So most of these secretions are actually peptides in structure, and they can be referred as regulatory peptides. So there are total 28 peptides that have gut regulatory functions. So all these substances that are released by endocrine and paracrine cells of similar morphology are called neurohumoral regulatory molecules. Now each type of endocrine and paracrine cell has a characteristic distribution within the gastrointestinal tract. For example, gastrin producing cells, which are called G cells, are found primarily in the distal portion of the stomach, and very few are found elsewhere in the gut. On the other hand, cholecystokinin or CCK producing cells are found in the small intestine, especially in the proximal region of the small intestine. Similarly, somatostatin and serotonin producing cells are distributed along the entire length of stomach. So, although endocrine cells are distributed throughout the gastrointestinal tract, the production of individual regulatory molecules may be confined to specific areas. So, in this extrinsic control system of gastrointestinal stimulation, sympathetic and parasympathetic gastrointestinal stimulation causes either the stimulation or inhibition of the gastrointestinal motility. In general, the sympathetic system is inhibitor of the gastrointestinal system, while parasympathetic system is the stimulator of gastrointestinal activity. Parasympathetic system is considered as rest and digest system, and it enhances the gastrointestinal activity of all gastrointestinal tract. So in this way it increases peristalsis, mixing movement, and glandular secretions in the gastrointestinal system. So parasympathetic system promotes the peristalsis and gastrointestinal tract activity, while sympathetic system is the inhibitor of gastrointestinal tract activity. Consider when you are not under stress and parasympathetic system is also working properly and food is also present in the gut, then this system will stimulate the neurons in the myenteric plexus of the smooth muscles and submucosal plexus also. And in this way, peristalsis will occur and submucosa will cause secretion as well as reabsorption. In this way, they help in the digestion of food. So parasympathetic system increases peristalsis, it increases the frequency of peristalsis and also increases its intensity. On the other hand, sympathetic neurons secrete a norepinephrine, which is an inhibitor of gastrointestinal tract activity, and they secrete this norepinephrine in the myenteric plexus. And when this decreases or inhibits the gastrointestinal tract activity, then the mixing and propulsive movement of peristalsis are also inhibited. So in this way, it will reduce the secretions in the gastrointestinal system. As we know that sympathetic nervous system innervates the organs of the body only when we follow the stress condition. In case of fight or flight mechanism, sympathetic fibers innervate the organs of the body. So in this case, sympathetic fibers also act directly on the blood vessels which are present in the gastrointestinal system, and when it will act on these blood vessels, it will cause their constriction, and this constriction is done so that the blood is redirected to other organs which need it more.